Great, wonderful to be here. Thanks for the invitation. So today I'd like to talk to you about FAIR and its application in terms of knowledge graphs and knowledge discovery. And the rationale here is that there's vastly more amounts of data available to us today than there ever has been. And the real challenge that we face is how to gain access to it and how to use it in an effective manner. And so really, you know, it's transformative because we don't need to necessarily collect the data ourselves, but we have to think about the research we want to do with other people's data. And so let me give you an example of this kind of work. This is work by a colleague of mine at Stanford, Professor Khatri, and he was interested in developing a gene signature for acute rejection, organ transplant rejection. And his strategy and the strategy of the team was to look through a database called the Gene Expression Omnibus. This is a database of gene expression data. Gene expression data shows you which genes are turned on and which genes are turned off under different circumstances. And so by searching this database, they were able to identify data sets that related to organ transplant rejection in heart, in liver, in lung, in kidneys. And what they wanted to do is learn a signature that was robust across these different tissues, these different organs. And so this gives you a little bit more power immediately with a metadata, a kind of meta-analysis, which will better estimate the effect of any one gene in this process. And ultimately, by doing this, they narrow the set of candidates to a set of a gene signature, which you see on the right-hand side, and they ask the question, well, what drugs could be used to modulate this particular gene signature? And they used existing drugs on the market that would target these things and see, can we extend the life, the survival of grafts? And they did this with a number of existing drugs on an animal model. And furthermore, what they said is, well, animal models are fine, but ultimately we have to apply this in the clinical human context. And so what they did was they accessed data from electronic healthcare records to see, are there people who have been taking these drugs and also have been exposed to these drugs, antidepressant drugs and so on. And they were able to find such individuals, and they could show that the lifetime, the survival of their grafts was extended for people who were taking these very common drugs. So this is a really nice example of, one, using other people's data to synthesize a new hypothesis about what could be involved, what mechanism is at play for graft survival. And secondly, to use electronic healthcare record data to validate that hypothesis for real patients. But if you ask Pravesh and his team, I mean, this is a nice piece of work, but where was the effort? And the problem, of course, is that it takes a lot of time and effort to go through the gene expression omnibus, to identify the relevant studies, to understand what was done, and to obtain those data and to repurpose them for their own use. And generally speaking, this is the kind of work that data scientists ends up having to do the most. So most data scientists have to find those data sets and make use, understand them, repurpose them, and ultimately then they spend time, a little bit of time, on the algorithm development. So this is a relatively unproductive task. If somehow we could reduce the amount of time that it takes for finding and reusing data sets, then we can make more discoveries and more effective discoveries. The other concern, generally speaking, is that this is only a symptom of a larger problem. In the reproducibility of landmark studies, in psychology, in pharmacology, in cancer, and in machine learning, we find over and over again it's difficult to obtain the results that have been published. And this is due to the lack of very specific details in the methodology sections, and an inability that even when you reproduce the study as it was indicated, you are unable to obtain those results. And so it's not surprising that for some time already, researchers, meta-researchers such as John Ioannidis, have identified that many of the research findings that are just original research findings are very difficult to reproduce and are optimally false. And so this is also problematic in the sense, and given the context of our general inability to translate original research results into clinical effectiveness, you see that it's about 6 or 7% chance that a drug or an intervention that enters into phase 1 successfully completes through the phase 4. And this is problematic, of course, because we have large numbers of people that are involved in these trials. We spend billions of euros trying to be successful here, and ultimately we fail in the vast, vast majority owing to both efficacy and safety reasons. So I'm going to argue that it's time for us to rethink how we perform and document our empirical research. And in particular, I believe that part of the problem is not just the inaccessibility of these original research results, but also the poor quality of the data themselves and the metadata that come with it. And this inability to understand what was performed and access it means that we cannot reproduce other people's studies, and we can't gain a confidence that these results are true and generalizable for other contexts other than the experimental one. And this leads to our translational failure. It's also increasingly obvious that we can't just rely on people producing content for other people, but in fact our work as data scientists is dependent on the ability of our machines to access this content and to make sense of it. And so this means that the content that we produce really needs to be made for machines and not for people. So we need a new social contract, one that's supported by legal and technological infrastructure to make digital resources, to make research data assets available in a responsible manner. This is important, of course, because for us as researchers in the health domain, we know that we have access to data, patient data, and other sensitive data that we cannot make simply open and available to everybody, but we need to take some kind of safeguard to make these available for research and for validation. And so this is the idea behind the FAIR principles. So FAIR stands for findable, accessible, interoperable, and reusable. And it's basically a paradigm that has emerged from people who work with data, who think about data, and who need to access it, to start producing content for the machines that we use in our research. The FAIR principles were published in 2016, and since then have become enormously influential in not just the biomedical space, but across different disciplines. They have been endorsed by the G20 and the G7. They're part of the European Open Science Cloud. And of course, they're required from Horizon Europe programs, and many other funders and journals require you to have data management plans and execute on making data available to others. Now, the FAIR principles have a number in this little box here, in box 2, identified the functional requirements that are expected of people who create this kind of content. Generally speaking, they deal with identifiers to retrieve all forms of digital content and knowledge for both the data and the metadata, that you have data and metadata of high quality, and that use shared vocabularies and terminologies and data standards, that you provide detailed provenance about how this content was created and other contextual information that are important in its potential reuse, that they are stored in qualified repositories, that they become available to others on a large scale, and that you also provide standardized terms of use so it becomes easier for innovation, for use by industry and other partners. There are a number of different ways in which FAIR data can be created, and these, again, come from the grassroots. Sites like RDMKit and FAIR Cookbook are collections of recipes to make FAIR data for different data collections. So I encourage you to look at that and see, for instance, if you have data on gene expression data, on mass spectrometry data, and so on, communities are coming together to define what those standards is. Which identifier systems should we use? Which data formats should we use? Which terminologies and vocabulary should we use? Which licenses should be applied to the content that we create? In our work, we also understand, again, that these data ought to be available to machines. So we have created infrastructure tooling to assess the technical correctness of FAIR implementations. These are so-called automated FAIR evaluation systems, which will analyze a given URL or a given DOI or something like that, a persistent identifier, and generate a report about what it finds at the site where this content is hosted. So this is one way to understand the machine accessibility of the information that is otherwise expected by the FAIR principles. Now moving on to knowledge graphs. So a knowledge graph is a graph representation of knowledge in which the nodes or the vertices represent entities, and you have relations to other entities through the edges of the graph. An important part of knowledge graphs is not that only that many of the relations are between individuals, but there are also relationships between individuals and types, and classes, and also descriptions amongst the classes themselves. Now, one of the aspects that makes knowledge graph representation so interesting for us in medical research is that we can link different knowledge graphs together. Now, this depends, of course, that the nodes, the identifiers that we use for the nodes and or the edges correspond across the different graphs themselves. And this is an issue of interoperability. But the idea is if you have one content provider, for instance, that provides information about a social network, people who know each other, and another one that provides information about travel, like the location of different sites to see in cities and so on, then you can start to connect these up. But of course, you need linking relations between these different graphs. Now, there are a number of centralized biomedical knowledge graphs that exist, but much more work is required to make them fair. And so you have resources like HEAD.io and the drug repurposing knowledge graph and many others that are emerging, and they are centralized integrated databases of facts that pertain to biology and medicine, information about genes, about diseases, about drugs and their activity and so on. These are super useful as research materials, but one of the problems is they centralize those data. And I think the idea, of course, is that all of you are creating content, and somehow we have to find a way to connect the content you're creating in a kind of global database. And so this is the idea that has been around for quite some time, the idea of the Semantic Web, pushed forward by Tim Berners-Lee. And the idea is that we create a set of standards to publish and share and query information on the web and using web infrastructure. And so this is intended to be quite successful, much in the way that the web has been doing, except instead of creating content for people to consume, we need to create content for machines to consume. And so together with many communities, we have been creating this linked open data cloud. Each of these circles are data sets, and the edges between them are relations across those data sets. They span many different domains, and including the biomedical domain, which is in the bottom right-hand corner. These comprise of ontologies and terminologies, and also data sets that we and others have put out on this linked open data cloud. One of our projects is called Bio30F. Here we have a set of data from over 30 different data sources, consisting of over 10 billion statements pertaining to drugs and drug action, proteins and domains, genes and genetic markers and animal models, and these are all interconnected. So this allows you to browse across these different resources to navigate from one database to another using a unified interface, which is the standards provided by the W3C. So much as you have hyperlinked web pages, we have hyperlinked data items. You can go from one resource in DrugBank to go to another resource in Kemble, in KEGG, and so on. And this is the idea is that we don't need to go to each different site, understand their APIs, understand their search boxes, but in fact, we can leverage all of this in a common infrastructure. Graph representations also allow us to examine data quality. We can start to look at the interlinks, the links between the different data sets, and understand whether they're consistent. We can examine links between DrugBank and PharmGKB and KEGG and see if they are consistent referent entities in that network, and identify where people are making mistakes or where the representations are different. So for instance, in some databases, stereoisomers of different chemical structures are represented as distinct entities, and in others, they merge them together. And so these are important from an automated knowledge representation perspective and also of integration of data, and they need to be considered in downstream applications. We can use these linked open data, these knowledge graphs, for a number of different purposes. We can create custom data portals. For instance, when the Ebola became a big thing some years ago, we created data portals quickly within a couple weeks' time in order to navigate potential drug targets and to understand what literature was around this. You can explore the content and create drug target disease networks and try to synthesize those elements that are going to be most promising for drug discovery. You can formulate interesting and sophisticated questions that require deductive reasoning, for instance, to understand what would be the potential impact of targeting a particular gene by looking at animal knockout studies to identify potential phenotypes of targeting that genes. And of course, we can use this information to build machine learning and deep learning applications to predict new drug uses. Perhaps an emerging topic of interest is the development of more sophisticated deep learning-based applications. These are so-called knowledge graph embeddings, where we try to learn the semantics of the information that we have in our graph by looking at their neighborhoods. And this, what you do is you represent the entities or relations with a vector, not just as a symbolic representation. And from these vector-based representations, you can compute similarities and also semantic relationships and allow more complicated vector operations. It also turns out to be quite effective in learning predictive models. And so the basic idea here is that for any node in your graph, in your network, you will learn an embedding or a representation of this node by its context by walking along the graph and building sentences that describe this entity. We can combine knowledge graph embeddings with other forms of embeddings, including from text representations, and together we can build these kinds of hybrid representations of what these entities are and how they're related to each other and use them in predictive tasks. I think important here too is that the newer generation of these kinds of algorithms are also looking at the aspect of explainable AI to try to understand the semantic relationships and to be able to explain the predictions that are being generated. Together with our colleagues in the Biomedical Data Translator project at the NCAT-supported project is to now try to do this large-scale data transformation and make available knowledge graphs from a wide variety of original data sources and that span across the different modalities as well as the different granularities of biology and medicine. And the idea is to build an information system that is intelligent in the sense that these entities are formally and semantically described, and we can build intelligent agents to navigate and recruit the information required to solve a particular problem. And that could be answering questions about rare diseases, making predictions about drugs for particular difficult and poorly understood condition, and generate new hypotheses about what investigators could be looking at. All of this is, of course, retrospective. We are trying to think about how to make data we've already generated available to machines, but we need to change that. We need to think about how do we generate content from the start that is already machine accessible. And part of the problem is that our incentive at the moment is to produce research articles in PDF format. This is not very useful from a data scientist perspective. It's useful from a person perspective. And then we use NLP algorithms to understand what is in those articles. What we need to do is think about the role of data not as secondary to a publication, but actually primary to the publication, for which we write a narrative or a story about our understanding, our interpretation of the research results. What we need to do is make data available immediately for reproducibility studies, to use in our own validation of our research results by using other people's data, and of course to create new hypotheses from. Together with my colleague Tobias Kuhn, we have launched a journal called Data Science, and the idea here in this journal is to develop methods and to promote the publication of structured knowledge from the start. This is what we call genuine semantic publishing, where the article is a database for which we write a narrative around. Already we have started, we have just published this one article which was automatically generated from a structured description of the main claims of the FAIR guiding principles, and many other researchers now are looking at their own articles and trying to publish structured representations of these, which immediately feed into our global knowledge graph. Good. Well, that was a rapid view of perhaps of FAIR knowledge graphs and the aspect of data discovery. I hope that I was able to describe to you what FAIR is about, and that FAIR really requires certain features for machine discovery and reuse, but ultimately offers new research possibilities that should reduce the effort of data scientists by using machines to help find specific data collections that are needed for their research, and also to understand the context about how these data were generated. And in total, by aggregating different research results, we become more confident about those research results. Fair knowledge graphs aim to provide this semantically annotated, machine-accessible, standardized, and AI-ready data, so you can spend more time thinking about the research problem and less time about how to structure those data. Together, AI technologies with these formal semantics enable us researchers to exploit an emerging internet of fair data and services in a semi-automated manner. And that really means that we can leverage machine agents to better find this content, to build models of it, and to make predictions that are of interest to us in biomedicine and health. And with that, thank you for your attention, and I'll be happy to take any questions at a later point and also at the coffee break. Thank you.